On, and thank you for joining Guggenheim during our second SMID-Cap conference. I am Debjit, one of the therapeutic analysts here, and joining me from CRISPR Therapeutics is CEO Samrat Kulkarni. Thank you so much, Sam.
Thank you for having us.
Before we get into the Q&A, a very quick overview of where you are and sort of the key milestones for 25.
Yeah, happy to do that. I think we've been around as a company almost 10 years, which many people don't appreciate and know about. But we've, ever since our founding, been focused on developing medicines that can transform the disease for patients with severe disease. And we started with what is now Casgevy, with sickle cell and thalassemia, and developed a transformative medicine that's on the market now in a commercial setting. But beyond that, I'm also very proud of the pipeline that we've built, and a very diverse pipeline. For those who are new to the company, we have a focus in cardiovascular diseases with in vivo gene editing, and where we take the CRISPR-Cas9 components encapsulated in a LNP particle, and we inject that into patients, and these LNP particles go to the liver and make an edit, reducing these cardiovascular risk factors. And I'll talk more about that. So that's one pillar that's continuing to expand. We have the best-in-class allogeneic CAR T. And so with CAR Ts, what we mean by that is taking cells or T cells from a healthy donor and engineering them and training them to kill a cancer. But we've also made additional edits using the CRISPR system that makes them autologous-like. So you get the efficacy of autologous CAR T without any of the side effects that autologous CAR Ts have, like ICANS. And obviously the convenience of having allogeneic off-the-shelf therapy. And that we're developing both in oncology indications as well as autoimmune indications. And the autoimmune opportunity is significantly larger than oncology. And we're in prime position on both as the best-in-class allogeneic CAR T for CD19. We have other CAR Ts as well in that franchise. And then beyond that, we have regenerative medicine, where we're developing iPS-derived cells for type 1 diabetes that can act as artificial pancreas. Eventually, the vision is to have many different organ replacements off of iPS cells. So those are all things we're doing. In addition, we continue to push forward on the platform with CRISPR Tx, and that's to do all sorts of advanced editing that enable us to open up more diseases for patients.
Thanks for that. Maybe we start on Casgevy first. How much visibility do you have into how the commercial launch is going? And there has always been a little confusion as to what the exact profit share is going to look like, or the flow through to CRISPR from Vertex.
The profit share is very clear. We have a 40-60 relationship. The Casgevy program started off as a 50-50 partnership with Vertex. At some point, we decided to change that construct to a 60-40, where Vertex owns 60% of the profits and we get 40% of the global profits. So that's very clear. And I think the way it's going to show up for us on our P&L is a single line item at this point, because it's a net cost. And you see this as a collaboration expense. And when the franchise becomes profitable, you'll see that as the net profits attributed to us become net revenues, essentially. So that's how you're going to see it flow through to CRISPR. And it'll all become clear as the launch continues to mature. But overall, I think we're very pleased with how the launch is going with the Vertex partnership and their leadership in commercializing this globally.
Is there a level where the program becomes profitable in terms of number of patients per year?
Well, it's hard to say. We haven't guided to when that is. But I think what you've seen, a few things that people need to understand about these launches that they don't understand with cell therapies. People look at the cost structure, and there was a note coming out about the cost structure of cell therapy launches broadly, and the numbers are very high, so you have to have relatively high revenues to make it profitable. What people don't understand is that's only a part of those expenses are what you need to have a commercial infrastructure and to sell the product. A large part of the investments that we're making right now essentially are to expand manufacturing capacity as well as to continue our trials. We're expanding the label not just across regions, but also in patient populations. So that's lifecycle management. So when you take all that away, you're still at a relatively attractive gross margin with these types of therapies that are priced at this level for rare diseases. So our hope is ultimately that the franchise becomes profitable in the not too distant future, and then ultimately that the company has a path to profitability in again not very long term, but we've said in other places that potentially by 2028 we could be break even as a company.
Awesome. So you sort of alluded to lifecycle management. Given busulfan is such a hot button topic, what are you and Vertex thinking about from next steps perspective,
Yeah.
trying to move away from toxic conditioning?
Yeah, we're very invested in developing better and better solutions for this patient population. Sickle cell and thalassemia have been diseases that we've known about for a very long time that have not had the level of investment in terms of medicines developed from big pharma for a very long time. And so here we have Casgevy, which is a remarkable medicine for these patients, and we continue to invest in next generation conditioning, which could be gentler for these patients. And so that what that would do is allow more patients to benefit from Casgevy and expand the addressable market, and ultimately to get to in vivo gene editing that would get to millions of patients around the world potentially as that becomes more scalable. With gentler conditioning, the way it works is we have our own efforts essentially. That we have an ADC where we acquired the antibody from another company. It's an antibody towards c-Kit or CD117, and then we have our own toxin that we've conjugated to that antibody, and so we're developing that for gentler conditioning. Vertex have said that they have other agents that they're developing, not just one, but multiple agents towards gentler conditioning efforts. And so we're going to put it all together and see what's the best, and ultimately bring that to bear for patients.
Got it. And talked about gene edits. CTX112, you had some interesting data at ASH. It has two sort of unique edits. Could you walk us through that patient population, why you think this was an update that the street largely missed, especially from a disease severity perspective and the amount of expansion that you got?
Yeah, I mean, I think, I would say that all the KOLs at ASH were very excited to see the data and reached out to us to say, oh, wow, this is spectacular. This is a target that we found through our large-scale screens. It's called Regnase-1. It's not a well-known edit. Okay, so we found this back in 2018 together with a different partnership that we had as well. And it turns out that this edit in T cells is very interesting. It keeps the cells more naive and more central memory phenotype, at the same time have more cytokines and more inflammatory discharge from the cells that improves their cancer killing capacity, essentially. So that's a very unique combination edit. And a couple of years beyond that, Carl June, who's the sort of the founder of all the CAR Ts, came up with their own analysis and their own large-scale assessment of what the best edits are for these CAR Ts, and landed upon Regnase-1 as the best edit to make these CAR Ts more potent. And which is very interesting that looking at thousands of genes, we all arrived at the same gene, and we have a lot of IP to cover the use of Regnase-1 in CAR Ts. But essentially, in our December update, while the N was small, it was only 12 patients, and we have subsequent data that we disclose beyond that. But the expansion of the allogeneic CAR Ts goes up to 50,000 micrograms per copies per microgram, which is never been seen before with an allogeneic construct. They all are in the 3,000 to 5,000 range, except for one-off patients that may be that may not have an immune system that's competent. So for the first time, you have something that's showing autologous-like expansion, yet you don't have ICANS like you do with autologous cells or autologous CAR Ts that are commercially available. And that's a very unique combination to get all the efficacy that you want without having the side effects, and you have the convenience of being able to dose patients right away. And that's going to come in playing on a big way in autoimmune diseases, for instance. One of the things that investigators tell us right now with autologous therapies in autoimmune diseases is that these patients are lymphopenic. You have to take them off of therapy for a couple of months to even get their T cells and collect cells for autologous cells, CAR Ts. It's just not an easy way to do it. If you had an off-the-shelf solution with allogeneic CAR T, and you can immediately dose those patients with lupus or myositis or scleroderma, it's a remarkable advantage to do it that way. And we now have tremendous data to show that a CAR T will have better efficacy than a bispecific or TCE. The bispecific or TCEs or naked antibodies are just not penetrating deep enough to have a very durable or full remission of the symptoms for patients suffering from these autoimmune diseases.
So for CTX112, what's the registration pathway going to look like? And have you started having this conversation with the FDA?
Yes, so we're pleased to have received the RMAT designation from the FDA for CTX112 in oncology. And so we're going to have that discussion on what the registrational path may look like in oncology, not in the not too distant future. So we'll update that. We promised the mid-year update on the program. So we'll guide the market on what that registrational path looks like. In autoimmune, we need to collect more data before we have those discussions.
So when do we get to see the autoimmune data, and what specific subtypes are you looking at right off the bat?
In autoimmune, we started the trial for lupus patients, but we've expanded the trial to a basket trial now, which includes scleroderma and myositis. So we want to include as many patients as we can before we do a data update. We've set mid-year for an update on the program, so we'll see where we get to on that.
So during mid-year, you have the regulatory update, you have potentially autoimmune, basket study update, and more likely than not, also cardiovascular programs?
Well, also mentioned oncology. So for 112, we'll have an update on, one of the questions is, yes, you're seeing remarkable data with 112, like autologous therapies, but what's the durability look like? And we want to show durability data as well in oncology, in the heme setting, in both indolent and aggressive lymphomas. So that's the other part of that update. And in cardiovascular, for both CTX 310 and 320, we said we'd have a first half update.
Got it. So let's switch to the cardiovascular side. Two separate programs ongoing right now.
Mm-hmm.
What kind of, as you dose escalate, where do you want to be, from as you get closer to the data sort of disclosures? How many cohorts, how many patients?
Yeah. I think for those who are not aware of these programs, we have two programs, CTX 310 and CTX 320. CTX 310 targets ANGPTL3, which is an enzyme which breaks down lipoprotein lipase. And essentially, if you inhibit ANGPTL3, you have more lipoprotein lipase to get rid of lipoproteins that are in circulation. So eventually, you have less LDL and triglycerides. So you'll see both triglyceride reduction as well as LDL reduction. The second program, CTX 320, targets Lp(a). This is sort of the third silent killer beyond LDL and triglycerides that people don't often talk about, but now there's a lot of awareness of Lp(a) because it's now sort of the explanation for why there are people who have very low LDL, reasonable triglycerides or in control triglycerides, are fit but still have MACE events or heart attacks in their 40s, sometimes early 50s. And Lp(a) is one of the reasons, which is a pretty atherogenic factor. In fact, it's almost 6x more atherogenic than LDL cholesterol, which you pointed out to Devjit in your reports. And you've actually been one of the few that have shown a light on Lp(a) and what an important target it might be in cardiovascular medicine. And so 320 targets Lp(a). And I think what we want to get to in both of these, first order is with our LNP platform, what dose level do we need to go to get the target reduction in either ANGPTL3 or Lp(a), and how much room do we have there at the top? And then, of course, what does the safety profile look like for a single administration of an LNP particle?
So, on the LNP, how much work has gone into? Do you have two unique LNPs for Lp(a) and ANGPTL3?
It's the same LNP platform.
And
so, yeah.
How much work has gone into that LNP? Because there was obviously a lot of noise around LNPs about a year and a half ago.
Yeah, so we've done a tremendous amount of work on the LNP. People often say, okay, where does the LNP come from? LNP systems are very complex. There's 4 components, sometimes 5 components. Some of the components are the common ones between different approaches that companies have taken. And then, the ionizable lipid is one component, but a lot depends on the formulation. You can formulate the same LNP components 6 different ways, different sizes, different densities, different solvents, et cetera. And that will change your attributes in vivo quite a bit. And you have to think about what fenestration do human cells have versus monkey cells or mouse cells. All those things come into play. So we have a very proprietary formulation where we did a lot of work, which gives us confidence that we'll have a very good safety profile for the LNPs while achieving the target reduction. But again, the data will tell.
And is the goal to be in that, let's call it 0.7 mg per kg range where Intellia kind of settled in, or can you shoot for something lower?
Well, I think we'll have to see where things land on the right dose level for expansion. Some of these factors depend on the guide potency, the availability for editing for that gene from a chromatin standpoint. So I think it's hard to say how do you go from monkey dose levels to human and how that may translate to humans. It may not all just be the same. In some indications it may be lower. In some places you may need to go higher on the LNP dose.
And let's stay with Lp(a) for a while. The push out for the Horizon study, does that complicate plans for the team CRISPR when you start thinking about what a registration study could look like? Or do you really need to wait for Horizon Lp(a) to read out to see the magnitude of benefit, et cetera, before thinking about what this could be?
Well, the Horizon readout is a very important readout to show that, yes, we've seen Mendelian studies, we've seen natural patient studies, epidemiology studies, et cetera, showing the effect of Lp(a). but does a pharmaceutical or a pharmacological reduction of LPA result in lower risk? That's the big question mark, and the Horizon study will show that, hopefully, and show that it's, across thresholds in that study. It is an important study for us to determine whether we would need an outcome study versus a biomarker-driven study for eventually a registrational trial. But I don't think we lose any time right now. I think that this year for us is more about establishing the right dose, expanding that dose level once we establish a dose, getting more sites opened, and getting ready for what could be the pivotal trial.
So you don't think you need an outcome study with Lp(a) or any of the cardiovascular targets, and it could just be sort of commercialized on a biomarker? Especially on the reimbursement side, for example?
On LPA, right now, we don't know. I mean, I think at this point, it hasn't been established, right? So the presumption is you need an outcome study. But if Horizon trial reads out positive and it shows that LPA reduction leads to better outcomes, then I think we could make an argument that you don't need an outcome study. Especially because, here's something that we know is 6x more atherogenic than LDL. And then how, as a regulator, would you ask a company or sponsor to say you put several thousand patients on a placebo when there are available therapies out there? That becomes very challenging, or you kind of lose clinical equipoise. So if the biomarker is established, and it's not a very complex biomarker, right? It's just LPA itself is the protagonist in creating these plaques and creating atherosclerosis, then it should be a much easier equation to say you don't need an outcome study. But all that depends on how Horizon reads out and Amgen study reads out over the next year and a half.
Got it. So just switching back to the allogeneic programs. So now you have 3 knockouts and 1 knock-in, right? Is that sort of the upper limit? Or sort of have you settled on the edits you need to do based on the data that you have at hand so far?
Yeah, so it's short answer is it's not the upper limit, because we now have another very elegant way to do editing ex vivo for CAR Ts, where we can have a single cassette that we can insert that can express effectively multiple knock-ins, and have multiple knockouts with hairpin RNA that are also inserted in. So you don't have any of the risks of translocation, but you can have single edit that can create a much more sophisticated construct, which may be necessary in the case of some solid tumors. For heme malignancies right now, for CD19, I think we have what's best in class allogeneic. So I don't know that we need to do much more in terms of editing at this point. But for solid tumors, I think we continue to create very scalable, easily manufacturable ways of editing that can incorporate even more sophisticated engineering in the cells.
Got it. Within the cardiometabolic franchise, you also have the AGT as a target. Mm-hmm. How are you thinking about that, especially given the permanency of the edits and the risk of hypertension?
Yeah, I mean, for those who are not aware, CTX340 targets a factor known as angiotensinogen, which is upstream of ACE, where the ACEs and ARBs work. So you're going, most blood pressure medications work either with ACEs or ARBs. And, what people don't realize is refractory hypertension is a huge burden across the world, but especially in the US. There's almost 1 million patients with refractory hypertension that are on 3 to 5 medications per day to manage their blood pressure, which is, oftentimes these patients are thrown off, because they take something else, they have an infection or whatever else, they take some other medication, there's some side effects. And it's actually one of the big causes of hospitalizations and mortality in this population with refractory hypertension. So what we're aiming to do is bring that baseline hypertension blood pressure level down, so then you can manage with one or maybe one other agent. You can titrate and manage it. The second thing is with zilebesiran, with the siRNA approach for AGT, you haven't seen many cases of hypotension, even at the very high knockdown levels, because I think the body has its own way of regulating the hypertension aspect, not so much the flares above. But I think first instance the goal is to bring that blood pressure down to a manageable level, and that I continue to be amazed by how using genetic markers we can control these very fundamental processes in life, which is maintaining blood volume and pressure is like one of the most important things for us to remain alive. And we can modulate that using angiotensinogen. And the other one that we showed data on recently in both monkeys and mice was in the eye, maintaining intraocular pressure. We were able to edit a protein called myocilin in the eyes, and we were able to reduce the intraocular pressure by several millimeters of mercury with one single edit in the front of the eye. And it's quite remarkable that we can do that. And similarly, I think with angiotensinogen, which we found KOLs to be extremely excited about as a very important unmet need that we can address.
Got it. So if I understand this program, then you go after treatment-resistant hypertension patients and try to get a, let's say, 10 to 12 millimeter reduction in mercury, and kind of you got to get to your goal. You don't really need to push dramatic.
And then, some patients, I think one thing that we may do that differently for angiotensinogen versus LPA or ANGPTL3 is potentially leverage the fact that we can multi-dose. So we may not want to get every patient to a 90% reduction with one dose on editing. What we've shown actually, and we haven't shown these data, but we've done experiment studies in monkeys where we can show that we can do multiple doses. And, 2 doses doesn't mean 2x the editing. It's like 1 point something. But you're able to titrate editing up to a certain level. So you may, for these patients, potentially interrogate a 2-dose regimen to get the editing exactly where you want it to be. And then in some cases, you may not need any other agents to manage their blood pressure. In other cases, you may have one residual agent where you can manage it up and down.
Got it. I wish we had more time to talk about the hypoimmune cells on the diabetes side, but maybe a very quick soundbite on that.
Yeah, I think, iPS-driven organ replacement is becoming bigger and bigger deal. I think the data from another competitor, Sana, showed recently that these cells are generally hypoimmune, islet cells, by the time they're differentiated. And if you 1 or 2 edits on top, you can make them very hypoimmune. And now we're like one of the furthest along with iPS-derived gene-edited hypoimmune islet cells. And so we're continuing to push that forward. In addition, we have cells that are embryonic stem cell-derived program, CTX211, in a device, which we've continued to push forward as well.
Well, awesome. Thank you so much, Sam. I appreciate the time today.
Thank you very much.
And good luck for 25.